Good morning. My name is Brenda Finucane. I'm a genetic counselor, also the Associate Director and Professor at the Autism and Developmental Medicine Institute of Geisinger Health System in Pennsylvania. Today I'm going to be talking about some family studies that Geisinger is doing in collaboration with the Simon's VIP project, and talking about particularly how we're using family studies to help predict and look at clinical variability for genetic copy number variants such as 16p1 1.2 deletions and duplications, 1q2 1.1 deletions and duplications, etc. So I'll start by saying I have no conflicts of interest to disclose for this presentation. And my topics for today will include a discussion of developmental disabilities and psychiatric disorders, just so we're all on the same page about what those conditions are. And then I'll talk about genetics and copy number variants and how changes in the amount of genetic material one has, whether it's a deletion or a duplication, can really affect the dosage of gene products and how that can contribute to clinical variability that we see when a child or an adult has a copy number variant. I'll talk about the implications of that for prognosis and intervention. I'll also talk about the things that we know about that contribute to the wide variability that we see in people who have different types of copy number variants. I'll talk about the importance of family studies and the studies that we're doing now to look at this, and also current and future research directions. So let me start by giving a definition of developmental disabilities. And this is a group of disabilities that's been called by a number of different names over the years. We also sometimes call these intellectual and developmental disabilities, also neurodevelopmental disorders affecting the brain. But one definition from the Centers for Disease Control and Prevention is that developmental disabilities are a group of conditions due to an impairment in physical, learning, language, or behavior areas. And they start during the developmental period. That means, from the time of birth or just before through age 18 or so. And they impact a person's day-to-day functioning. And they're often chronic diseases typically, and they last a person's lifetime. And it's estimated, according to this broad definition, that 1 out of every 6 children has a developmental disability. So that's a lot of children and a lot of adults, as you can imagine. And so on this slide here, you'll see there's lots of kids with different types of developmental disabilities. These can include intellectual disabilities, cerebral palsy, autism, for example. And in this case, on this slide, these kids have different genetic causes for their developmental disabilities. So some examples, as I mentioned, are intellectual disability, or ID, ASD, or autism spectrum disorder, CP, or cerebral palsy, a specific language impairment, learning disabilities, attention deficit disorders, epilepsy, and the list goes on and on. So I'll switch a little bit and talk about psychiatric diagnoses. And generally, people tend to think about psychiatric diagnoses as adult onset conditions like schizophrenia, bipolar disorder, depression, etc. These can be both childhood onset, like autism, which is a psychiatric diagnosis, or adult onset conditions like schizophrenia. But what are psychiatric diagnoses? In essence, they're observed, recognizable patterns of human behavior. That's what a psychiatric diagnosis is. They're based on these patterns of human behavior, and that's how they're defined. And the criteria for psychiatric diagnoses are laid out in a big manual called the Diagnostic and Statistical Manual that gets updated regularly. We refer to this as the DSM. The most important thing to remember about psychiatric diagnoses, because I know for parents, particularly of young children, just the term psychiatric is a very scary thing. There's a lot of stigma related to psychiatry and psychiatric illness. But really, in the end, what we're talking about are conditions that can happen in childhood or adult life, and they're symptom diagnoses. So they're defined by their patterns of behavior and their onset and some other criteria that we can observe. They don't emphasize the etiology or the cause. And when we talk about a genetic condition like a chromosome deletion or duplication, those are cause diagnoses. So much of what I'll talk about today are the differences between symptom diagnoses and behavioral diagnoses versus cause diagnoses. And it's important to know that psychiatric diagnoses are not diagnosed using laboratory tests. They're diagnosed based on observation. So there's no MRI signal that will diagnose schizophrenia or autism or intellectual disability. The brains of most people with intellectual disability on MRI look very much like the brains of anybody else in the population. Really, there are other types of tests that we need to do to measure behavior, et cetera, rather than laboratory tests. Now, you'll know that I mentioned autism as a psychiatric diagnosis. It's a childhood onset psychiatric diagnosis, but I also mentioned it as a developmental disability. So you can see that many developmental disabilities are, in fact, psychiatric diagnoses. They're childhood onset psychiatric diagnoses. So here's a list of a number of psychiatric diagnoses, ADD, anxiety disorder, et cetera, which are also diagnoses that many children have and can make them eligible for special education services, and certainly would also, many of these come under the heading of developmental disabilities. So I mentioned etiology, and let me talk a little bit more about that to try to distinguish different types of diagnoses. Etiology means the underlying cause of something, and so developmental disabilities and psychiatric disorders are symptom diagnoses, as I mentioned, and we now know there are numerous different causes. We didn't know this many years ago because we didn't have the genetic tests available that allowed us to see the causes, but now we can see many of them. And we know that genetic and or medical factors play a role in causing most developmental and psychiatric disabilities. It's important to note here that that doesn't discount the importance of environment. School is really, really important. Therapy is important. Medications are important. Things can really impact the way these developmental disabilities and psychiatric disorders manifest and how we manage them. So just because we're saying there are genetic and medical factors that play a role here does not mean that it's set in stone that a particular disorder is going to manifest in a certain way. There's a lot we can do to influence the way these are manifesting themselves. In fact, we can't control the genetic factors, but we can control environmental and medical interventions. If you look at this slide, what you'll see is that we're now able to diagnose many genetic causes of developmental and psychiatric disorders. So if you look at this pie, you can see that CNVs, or copy number variants, which I'll describe more fully in a little bit, these are deletions and duplications of chromosomal material. We know that if you do a microarray analysis, which can detect these differences in chromosomal quantity, that about 15 to 20% of people with an intellectual disability will have a copy number variant. In autism, it's about 10 to 15%. In epilepsy, about 9%. Schizophrenia, it's about 5%. And now we have an even more powerful test known as whole exome sequencing, which actually looks not just for differences in the quantity, but changes in the genetic code. And now when you look at this population of individuals who have developmental and psychiatric disorders, and you're using microarray and also using whole exome, about 40% conservatively can be identified as having a specific genetic cause. And that's really powerful. Years ago, when parents would come and ask, why has my child got developmental delay, or why is my child behaving differently? We really didn't have a lot of ways of answering that. We could look at the symptoms and diagnose it, but we couldn't really find the causes. We're now really in a very good position to identify causes. And of course, the Simons VIP is attracting families who have children or who have themselves known causes of these disorders. So I will walk you a little bit through the genetics of this so that we can define what copy number variants are. And if you look at the top of this slide, you hear the word genome, and genome refers to the sum total of all of our genes. And these are in the cell, and they're packaged into these structures called chromosomes, and chromosomes contain genes. And the genes ultimately in our bodies and every one of our cells are the chemicals that contain the instructions for making proteins. And proteins are what genes do. They make proteins, and the proteins can act alone, or they can form other types of chemicals, and they perform all of the things that our bodies need, whether it's to form a particular structure when a baby is developing and before birth, or even during life. Certain chemicals that are expressed in the brain are produced due to genes. So if you look at chromosomes in the cell, they're all sort of scattered around like this. They look like little sticks with lots of different stripes on them, or bands as we call them. And the ability to look at chromosomes like this has been around for decades. We have been able to look at chromosome pictures literally since the 1950s. So when we look at these chromosomes, we're seeing these structures that contain many, many, many genes. We're not able to look at the individual genes here. We're really just looking at the packages they come in. And I've lined up the chromosomes here just to show you that if we took those chromosomes and straightened them out somewhat and lined them up, and on the left you'll see there's a drawing of the chromosome if it was perfectly straight and showed all the stripes or bands. And on the right is an actual picture of a chromosome. And you can see they come in different sizes. They come in different patterns of stripes or bands. And they're given different numbers, and these numbers were given many, many decades ago so that we could be able to know what we're talking about. So when we say a child has a 16p11.2 deletion, for example, what we're talking about is the 16th chromosome and a particular region on that chromosome. So if we took out a chromosome, and I'll give you an example here of chromosome 3, we have different names for different regions of the chromosome. And so for chromosome 3 and for all the chromosomes, there's a part called the centromere, which is kind of a pinched in area of the chromosome. We like to think of it as a waist. So a waist, and W-A-I-S-T, so a little narrow area of the chromosome. And above that waist or centromere is a smaller piece of the chromosome that we refer to as the P arm. And that P came from the French petit when they first described chromosomes, and that was the word for small. And then Q follows P in the alphabet, so they called the long arm of the chromosome the Q arm. And so when you hear about a 1q2 1.1 duplication, for example, they're talking about chromosome 1. Q says there's a problem or a duplication on the Q arm, the long part. And then as you can see from this slide, there's these different regions depending on the stripes that you can see under certain conditions in the laboratory. And so there's a region called region 2, and then there are subregions. In this example, we're talking about chromosome 3p22. And what that means is we're talking about chromosome 3. We're talking about the P arm or the small arm. And in that small arm, we're talking about region or band 2, and then the subregion 2. And so when you say 16p11.2 deletion, it's really not 16p11.2. And you can impress all your friends by saying it correctly in the genetics way. It's 16 chromosome, P is the small arm, 1 is the band, and then 1.2 are the subregions of that band. So I bring this up because people are always confused why we have these conditions now with these very specific and strange number names. And it's got to do with the nomenclature or the naming of different regions of the chromosome. So that's your background that you need to understand the copy number variants, or CNVs. Copy number variants are differences in the number of copies of one or more genes or sections of DNA. So basically what we're talking about are deletions or duplications. So differences in the quantity of genetic material that we expect to see on the chromosomes. Now sometimes there are very large CNVs, large deletions and duplications that have been known for many decades because we could see them on a chromosome analysis. They were big enough that we would see the chromosome was short, it was missing a whole section. And there are conditions like Smith-Magenis syndrome, for example, that were known because they could be seen on a chromosome analysis. However, those CNVs, the really large ones, were not very common, and we didn't really have the technology until just about a decade or more ago to be able to really see very small deletions and duplications or copy number variants. And microarray analysis is a laboratory type of test that allows us to see very, very tiny deletions, duplications. We can sometimes refer to these as micro deletions or micro duplications because they can only be seen using an analysis that gets to detecting them under the level of the microscope. If you were actually to look at the microscope at the chromosomes, they look perfectly fine. The deletions and duplications are too small to be visualized. So many, many new CNVs have been discovered over the last decade because of microarray analysis, and the custom now is to diagnose or name these new CNVs using the chromosome number, the arm, and the area where they're deleted or duplicated, which is why you have those difficult names sometimes for your children's or your own conditions. I've heard many families say, why can't we just have Smith syndrome or something like that instead of calling it a 16p11.2 duplication? Well, there's reasons for that, and you're in good company because there are literally hundreds and hundreds of different CNVs that have now been discovered through microarray analysis. An important thing to know is that CNVs often involve multiple genes. So there may be many different genes involved that have been deleted or are additional. And in genetics, if you have too little genetic material or too much genetic material, many, many times what will happen is the blueprint is altered and you'll see differences in the way a child develops physically, might look different than other children or the way that child would have looked if he didn't have the deletion or duplication. And it also may affect the way the child develops behaviorally, intellectually, and also may cause some physical or medical problems in a child because of the difference in the amount of genetic material. So we refer to that as gene dosage. And so if you have too little genetic material, you may not be getting enough of the protein that those genes are supposed to form. And if you have too much genetic material, if you have a duplication, let's say, you may be getting too much of a gene product or overexpression. We refer to CNVs as pathogenic, benign, or variants of uncertain significance, or VUS. Pathogenic CNVs are ones where we know that they can cause a problem. And so for the 16p11.2 deletions and duplications, for the 1221.1 deletions and duplications, we know that those particular CNVs consistently can cause problems, although there's wide variability. There are others that are just found in the general population, and they're really not something that causes a problem for people. And those are referred to as benign. And then there's a lot of them now that we're not quite sure. You may have a child who's having some developmental problems. They have a microarray analysis done, and then a CNV comes up. They have a little bit extra of genetic material, but it's not really been reported in other children with developmental problems, and it's not really clear whether it's benign or whether it's causing the child's problems. So we call those VUSs, or variants of uncertain significance. Today I'm really going to focus on pathogenic CNVs, ones that we really do know are associated and causative of certain symptoms, whether they're behavioral, developmental, or physical. The other important thing to know is that CNVs can be de novo, meaning they occur out of the blue in a child, neither parent has the CNV, or sometimes they can be inherited from a parent. And so those are two different ways that we see CNVs happening. Some CNVs that can cause very severe symptoms behaviorally or intellectually, you don't see them often running in families because it can really impair the person's ability to have children in the future, et cetera. So it may occur out of the blue and then it's not passed on in a family. In other cases, you may see a very mild manifestation of a CNV in a parent who may not ever have known that he or she had this CNV until they had a child who was diagnosed with it. And maybe the child has more significant problems, and then they go back and check the parents and say, oh, goodness, dad has it as well, but he really didn't have those same problems. So here are some examples of copy number variants, and if I was actually to list every single one known, it would take slides and slides and slides because there are hundreds and hundreds of CNVs that we know at this point. One of the things that has happened in the world of developmental disabilities is that many children now are getting genetic testing and they're being identified as having CNVs or other genetic disorders, and there's a lot of confusion because, as I mentioned, we have names for lots of developmental disabilities and psychiatric disorders, these symptom diagnoses, but now we're also finding cause diagnoses. So, for example, here are 3 kids in a preschool, all diagnosed with developmental delay, autism spectrum disorder, and all 3 have been said to have ADHD or attention deficit hyperactivity disorder. But if you do genetic testing, what we find is the little boy on the left has a genetic condition called Fragile X syndrome. The little girl in the middle has a duplication 15q, and the little girl on the right has the 22q11.2 deletion syndrome, which you now know these describe different chromosomes and different regions. The same is true in school age. So we have 3 kids, let's say, in a learning support classroom in a school. All 3 have an intellectual disability. All 3 have been diagnosed with obsessive compulsive disorder, and all 3 have an anxiety disorder. I often think they're probably anxious because they have too many diagnoses. But in any case, if you did genetic testing, what you'd find is the little boy on the left has the 22q11.2 deletion. The girl in the middle has something called Williams syndrome, which is caused by a deletion of chromosome 7. And the little boy on the right has the Smith-Magenis syndrome, which is caused by a deletion of chromosome 17. So what's going on here? Well, what we know is we've got genetic laboratory technology advancing to the point where we can find causes, but then kids are still appropriately getting diagnosed with symptom diagnoses for their disability. And so I often hear from parents, well, if my child has autism, but now we know he has a 16p11.2 deletion, should they drop the autism diagnosis? The answer is no. The autism diagnosis just describes the symptoms that you're seeing on the outside, and that's really helpful. Knowing about the 16p11.2 deletion tells you what the cause of those symptoms are. So it's like apples and oranges. So the important thing to think about in terms of the genetics and the CNVs and this psychiatric and developmental diagnoses is that the genetic causes, or the CNVs, are the reason for the behavioral and intellectual symptoms that get classified as autism or intellectual disability. So instead of saying my child has autism and a 16p11.2 deletion, what's really more technically appropriate is to say my child has autism because of a 16p11.2 deletion. So what do we know about CNVs, including 16p11.2 deletions and duplications, 1q21.1 deletions and duplications, and all the others? Well, we refer to something called a phenotype when we're talking about the way these gene problems manifest themselves in children and adults. A phenotype describes the observed characteristics related to gene expression. So if you have too much or too little genetic material, and if it's known to cause a problem, that problem might be very variable, and we would describe the set of problems that go along with that gene expression as a phenotype. I'll give you an example that's well known to most people around Down syndrome. So most people, even in the general population, can recognize somebody with Down syndrome. It's not because everybody with Down syndrome looks exactly the same. They certainly don't. They're just as varied as anybody else in the population. But they have a key set of physical features that's recognizable. That's part of their physical phenotype. There's also something we call a behavioral phenotype, and the behavioral phenotype describes the expression of learning and behavioral characteristics. As you can imagine, learning and behavioral characteristics also have to do with the environment, with school, with culture, and other factors that are not related to the expression of genes. So it's a very complicated thing when we start thinking about behavioral phenotypes. But many of the underpinnings of behavioral expression and learning abilities really come from the genetic underpinnings. many of the behavioral and learning characteristics that we see in the human population come from our genetic underpinnings, and of course there's an interaction with the environment. We know that the clinical symptoms or the phenotypes of CNVs are in many ways related to gene dosage. Is it because you have a deletion and you're not getting enough of a certain gene expression, or do you have too much? You have a duplication and getting an overexpression. And of course, genes all interact with other gene products, so it becomes very complex. And there's wide variability in the clinical phenotype, whether we're talking about behavior or physical problems, from one person to the next, even with the same CNV, even within the same family. So you may, as I mentioned before, have a child who's diagnosed with a 1q21.1 duplication, let's say, and has a cardiac defect and also has some significant developmental delays. And then when we do testing, we find out that mom also has that same CNV, but really had relatively few problems and certainly wasn't born with any kind of a cardiac or heart problem. And this slide shows for many of the most common CNVs that we see and that we know are pathogenic or causative of problems, that you can find them in a lot of different groups. So, for example, in kids or adults who have blood samples done for microarray for intellectual disability or some kind of developmental delay, we find all of these CNVs that are listed on the left side, these deletions. But let's say you had a group of kids at an autism clinic and they did microarray analysis and they decided to look at them. Well, you would find the very same CNVs in kids who have an autism diagnosis. If you looked in adults with schizophrenia, for the vast majority, you would also find the very same CNVs in schizophrenia, and the same is true of epilepsy. So what this tells us is that the very same CNV can show itself differently and result in different psychiatric or developmental disorders in different people. And this becomes really a source of stress, not just for families, but also for professionals, because let's say you have a baby who's born and diagnosed with a CNV that we know causes a wide range of problems, either physical, developmental, and psychiatric. But we don't have a crystal ball, and we can't figure out, okay, so this baby in 20 years is going to develop schizophrenia, or this baby with this CNV might have just minimal learning problems or a few problems at all. And so it has implications for prognosis. In other words, trying to figure out how to predict what the variability will be or what the particular manifestation of a CNV will be for any particular person. And of course, that's important for intervention, because if we knew this CNV in this child will cause autism features, then we would want to get in there and really try to do intensive therapy to see if we can help with those autism features before they really manifest. But the fact is, because there's so much variability, we can't predict in that way. So we know that 16p11.2 deletion, as an example, is associated in a minority of individuals with autism, so problems with social impairment and communication, and it's a developmental disability. But most people who have a 16p11.2 deletion do not have autism. So again, let's take that 1-year-old who just gets diagnosed on microarray with that deletion. Wouldn't it be nice if we could figure out which of those babies was actually going to be in that group that develops autism, so that we could really customize and maximize that intervention. This causes stress for families. It causes stress for professionals. I can tell you as a genetic counselor, it's also stressful for us not to be able to give any clear guidance. It makes us feel like, we're telling you, well, it could be anything from completely unaffected and a person who has a very typical life and somebody who needs very, very intensive supports. So it's really not adequate right now because we just don't really know enough about the variability. So what contributes to phenotypic variability in people with CNVs? Well, there are many different factors. Today I'm going to focus on family background genetics and some of the work that Geisinger has been doing with Simon's VIP around family background. But obviously there are other factors as well. So, for example, a baby who has a CNV but who's also born very premature may have an impact from that prematurity also that compounds some of the features. What about medical impacts? We know that children who have the 22q11.2 deletion are often born with congenital heart defects, and they often require surgeries, not only for their heart, but sometimes for the palate, the roof of their mouth, etc. What impact do those surgeries have on the development of a child who has to spend a lot of time in a hospital? What about the emotional functioning of a child who's growing up spending lots of time in doctor's offices and in the hospital versus a child with the same 22q11.2 deletion who doesn't have those problems and really has a very different experience? Obviously, that's going to have some differential impact. What about intervention and education? Of course, these are contributors. We know that, even great intervention is not going to cure a CNV or cure the impacts, but it can certainly help to mitigate some of the behavioral issues and really help foster good education practices in children, such as study habits, etc., that might really enhance their success in school. There are also social and cultural factors that contribute to phenotypic variability. I will often give the example of, we have kids born today who have a CNV, let's say, that might reduce their IQ or their intelligence quotient somewhat compared to their family or where we would have expected them to be. Now, 100 years ago, if they were living in a non-technical society, they may be able to do quite well because people really weren't measuring IQ, or they didn't have to do a lot of things that really impacted their IQ, and they had a lot of other skills that would allow them to live a very normal life. Today, it takes a lot in our modern society to be able to just navigate, the ability to use an ATM, the ability to drive, etc. All of those types of abilities put more pressure on us to have higher intellectual functioning and abilities. So we may look, if we have lower abilities, more disabled in today's society than we would have in another society. So those social and cultural factors are really important. There are also maybe environmental contributors. For example, lead exposure is known to reduce IQ in populations of kids who are exposed during the developmental period. So imagine you have a child with a CNV but also is exposed to lead in the home. That might be a double whammy, if you will, toward really impacting that child's development. I'll talk about two different concepts in genetics relating to the phenotype. Again, the expression of the characteristics we see in CNVs and how these describe variability. So there's two different concepts that we often talk about in genetics. One is variable expressivity. And what this means is that for any genetic disorder, whether it involves a developmental disability or not, there's often a range of the phenotype from mild to severe in individuals who have the same cause. And an example is we know in 16p11.2 deletion there may be people who have an intellectual disability and require lots of supports to people with above average intelligence who have 16p11.2 deletions. So there's a range of abilities. And this is not just for CNVs. This is true generally in genetic disorders. There's always a range. You may have more severe symptoms versus others. And that's what we call variable expressivity. So the gene is manifesting itself. It's producing symptoms of sorts, but some of them may be so mild that they don't cause a lot of problems, while others may be causing lots of problems. A different idea is what we call incomplete penetrance. The idea that some people with a copy number variant or an SNV or single nucleotide variant, as it's called, other types of genetic disorders, might not be affected at all. So it's kind of an all or none. Some people might have manifestations and others don't. An example of this would be we know that there are children with Down syndrome who have heart problems when they're born. They may be born with a hole in the heart or some other type of problem with their heart. And almost half of all babies with Down syndrome is born with one of those heart problems. But the other half are not born with those problems. So it's kind of all or none. And so that's what we call non-penetrance. And we would say that the cardiac or the heart problem in Down syndrome is penetrant in about 45% of babies with newborns with Down syndrome, but it's not penetrant in the rest. So it's kind of an all or none. Now that's different from saying mild versus severe, because we're just saying it's either there or it's not there. So when we think about developmental disabilities and psychiatric disorders, there's really a trend now, and certainly Geisinger and Simons are really pushing this idea and at the forefront of this new kind of way of thinking, that phenotypic variability for developmental disabilities and psychiatric disorders really is going to depend on whether you're talking about variable expressivity versus non-penetrance. So an example would be something we call a dichotomous trait or categorical diagnosis. So I told you before, autism spectrum, for example, or intellectual disability, these are symptom diagnoses, and there's criteria in that diagnostic and statistical manual for these diagnoses. And really, you either have the disorder or you do not. So you have autism according to the criteria or you do not. Many parents may have been in this situation where you took your child to a psychologist or a developmental pediatrician and they did lots of symptom-based testing on development and said, oh, your child has autism or meets criteria for autism, or your child does not meet criteria. So it's an all or none. However, we do know that there are a lot of kids who have a lot of autistic features. They may have things like hand flapping, or they may not be making good eye contact, but overall they're not meeting full criteria for an autism diagnosis. So in other words, it may not be quite all or none. It could well be, and what we believe is that there's a whole continuum of traits, and we set a certain cutoff for autism based on how many of those traits you have and other sorts of variables. So we're interested in looking at what we call quantitative or continuous traits, things that aren't all or none. Dichotomous means kind of all or none, one or the other. Continuous means there's a variability across a whole spectrum of people, and everybody's somewhere along that spectrum, but some may be closer to having lots of features that could get diagnosed as one of those clinical disorders like autism or ID. So quantitative traits have a continuous distribution in the general population, which I'll talk about in a minute, and we're really interested in looking at quantitative continuous traits that involve intellectual disabilities, behavioral differences, motor performance, etc. So quantitative traits are measurable characteristics, and they depend on the actions of many, many genes and the environment, and it's really your background genetics and your environmental experiences that lead to you having these traits. Everybody in the population has some of these traits. You know, everybody has a certain height in the population. Some people are tall, some people are short, but there's a whole continuum of where you are on that height curve, for example. And they vary among individuals over a range, and they produce a whole curve, a continuous distribution, which I'll show you in a minute. So examples include height, weight, blood pressure, certain personality traits, motor skills, and learning abilities. Everybody in the human population is somewhere along the curve for those particular features. Here's an example of a curve for adult male height in the U.S. population. And so here's the height in inches, and you can see that most males in the U.S. population are clustered around the average height of just under, 6 feet in the population. But you do find people who are very short, and you do find men who are very, very tall. We all know this. So that's a continuous distribution. Basically, we all are somewhere on that curve. And then on those curves, there's usually a big hump in the middle where most people cluster. But then you're always going to have people on the edges of those curves. We know that quantitative traits are highly heritable. They tend to run in families. So I show this picture because Michael Jordan here is 6'6", and skier Lindsey Vonn is 5'10", which is pretty tall for a woman. So both of these individuals would be on that tail end of the curve for height because they're much taller than the average. So if Lindsey Vonn and Michael Jordan ended up having a child together, and I don't keep up with the contemporary news and who's dating whom, so I don't even know if that's out there in the world of possibility, but let's just imagine that they had a child together. We would expect that their children would be tall. I don't think that's a leap of the imagination. We all kind of think in that way. I can also say, because they're both star athletes, and because motor abilities tend to be heritable as well and are also quantitative traits, that if Michael Jordan and Lindsey Vonn had a child, that child we would expect might have better than average motor abilities. So imagine Michael Jordan and Lindsey Vonn did have a child, and that child had a CNV that caused short stature. Well, that CNV might cause short stature, but that child was going to be tall to begin with just based on the background genetics. So maybe that child would not be as short as other people with that CNV. What if that child had a CNV that impacted motor function? Well, because that child from background genetics would be expected to have better motor function than the average child, maybe that CNV's impact would not be as bad on that child because the starting point was already going to be higher. That explains a lot of the variability that we see, that what you come to the table with, your background genetics, is kind of your starting point, and we all come with our quantitative traits at different starting points. So we're very interested in studying families to look at these quantitative traits, and that's why for those of you signed up with SVIP, we ask you to regularly complete these family studies, not just on a child, let's say, who's diagnosed with a genetic condition, but also on family members who do not have those genetic conditions, because it's by looking at families that we really get the true picture of the variability. So SVIP and Geisinger ADMI, our Autism and Developmental Medicine Institute, are collaborating on projects related to studying quantitative traits that influence learning and behavior, and also other things in the particular disorders that we're looking at. So for example, in the 16p11.2 deletion, we know that it can be associated with obesity. And so we want to look at family weight overall and see, we explain the variability in the weights of children who have this deletion. We're looking at the impact of family background on symptom variability, and we're particularly interested in people who have de novo CNVs. In other words, neither parent is affected, and it's just a child who has the CNV. Because when we look at parents and siblings who don't have the CNV, we can get a better picture about what's the family background and what are the quantitative traits that are running in that family. And then what was the impact of the CNV in that one child on functioning. We do this through online assessments of these traits, either in person or by online measures, as I mentioned. We do this using online assessments of quantitative traits in the person who has the CNV, but also in unaffected family members. And so a lot of you have been prompted to fill out these online assessments. And I know sometimes it can be really tedious, and it's really important to do because it's important work to kind of look at the whole family and to better understand the impact of these copy number variants. We're also looking at DNA analysis of the unaffected parents and siblings and the person with the CNV to look at that background genetic variables that contribute to the symptom variability. And so I'll give you an example of one of the tests that we often ask you to do online, and it's known as the SRS, or the Social Responsiveness Scale. And social responsiveness, this measures your responsiveness in different social situations and as a quantitative trait. So everybody in the human population is somewhere along this curve of social responsiveness. We all know people who are kind of extroverted and very socially skilled and can go into a cocktail party and just make friends with everybody and talk about a variety of subjects, and they have no anxiety about doing that. So they might be very much on the far left end of that social responsiveness scale. I'm not on that scale on that end. I tend to be closer to the right side of the scale. Many of us also know people like that who really are more introverted, maybe a little shy, have some social anxiety, and really don't have kind of the innate skills for social interaction. And then most people are right in the middle in that hump. It's just like height, weight, other things like that. You know, a lot of people clustering around the middle, some people on either side. On the SRS, the higher you score on that measure, it relates or correlates with decreased social responsiveness. So the higher you score, it shows us you have fewer skills in the social responsiveness area, or you're less socially responsive. And toward the very far ends of that range, what we find is that correlates with diagnoses of autism. You can't get a diagnosis of autism by doing the SRS, but what we do know is that in individuals who have autism, they as a group tend to have very high SRS scores. So what does this mean, and what do family studies mean for this? Well, let's look at this SRS, and remember that the higher you score in the SRS, it means that you've got decreasing social responsiveness. So in blue here, we see a family, and they have 4 kids, and parents do not have a CNV, and they have one child who doesn't have a CNV, but the little guy in orange has some genetic variant or copy number variant. And what we see is that the 3 other individuals without the variant score kind of right around that middle range of social responsiveness. But the child who has the genetic variant actually has been shifted, and his scores are further along or further down that scale of social responsiveness. And that means that the CNV, or the genetic variant, has shifted that functioning. It impacts that functioning in this child. Well, let's look at family B, and let's say we're talking about exactly the same CNV. Let's pick 1Q2 1.1 deletions. So in this family A, this is a child with that deletion, and we see that this CNV has caused him to have problems with his social responsiveness. In family B, you can see the starting point for the family is much further to the left. So mom, dad, and the child in blue all score kind of far to the left, meaning they have exceptionally good social responsiveness. They're not clustered around the middle like most people are. They actually are really on the more functional end of that and have increased social responsiveness. And because that's a highly heritable quantitative trait, that means that their child with the 1Q2 1.1 deletion has the very same impact on social responsiveness, but you can see that now that child's score is really within kind of very close to the normal range, which tells us that some kids with a 1Q2 1.1 deletion may end up having autism and some may not. And some of the influence of that has to be their starting point, what they got in terms of background genes from their family. We actually looked at this in a study, and thanks to the families who participate in the Simons VIP, we were able to get this data, and our group published a study on social responsiveness and other quantitative traits in the 16p1 1.2 deletion. There's the reference below. It came out in a journal called JAMA Psychiatry. And what we showed was this very thing, that you have on this graph siblings and parents, unaffected siblings and parents, of individuals who have a 16p1 1.2 de novo deletion. So it occurred out of the blue in this family. And if you graph the curves of the individuals on this, based on the data that was collected by Simons, what we see is that the curves for the parents and siblings are just about the same. There's really no statistical difference between those blue and green curves, and really they correlate with the general population curve. But when you look at individuals who have the 16p1 1.2 deletion, there's variability. They have their own curve, but their curve is shifted to the right. So the average person with a 16p1 1.2 deletion has decreased social responsiveness on the SRS compared with the average in the general population. What was interesting in this study was that we did show that the siblings and parents who were more on the left side of their own curve tended as a group to have children who, with 16p1 1.2 deletion, were shifted more to the left side of the 16p curve, meaning they had fewer social impacts. The impact was actually the same from the copy number variant, the 16p1 1.2 deletion, but because the starting point, because of background genetics, was higher in those families, their kids did not manifest as many symptoms. We did the same thing in this very study for intellectual disabilities and intelligence. We looked also at obesity, since that's part of the 16p. So let's look at IQ as a quantitative trait. So intelligence is a quantitative trait, meaning that everybody in the population is somewhere along an IQ curve, and the vast majority of people are really clustered around that IQ of 100, which is, quote, normal intelligence. Some may want to debate that because there's different kinds of intelligence, as we all know. But the type of intelligence that we measure on IQ tests really shows a nice curve like that. And really, we have people on the fringe edges, which some have very, very high IQs and might be considered gifted. And then you have a small percentage on the other side who have decreased IQ and might meet criteria for an intellectual disability. So on this slide, you see the black curve is for, quote, intelligence in the general population, and the average IQ for normal intelligence is around 100. And then there's criteria for saying when somebody has an intellectual disability. An intellectual disability is the name we give, or ID, for something that used to be called mental retardation. And it's been defined by people who make this definition that it's associated with an IQ of 70 or below. So 2 standard deviations, or this much of a distance between the average of, quote, normal intelligence and the end of the curve. The red curve here shows what the curve looks like if you just did it on IQ tests on people with a 22q1 1.2 deletion, which is one of the most common CNVs that we see. And what we see is you have plenty of people with a 22q1 1.2 deletion that have IQs in the, quote, normal range of intelligence, but we also see some down in the range of intellectual disability with an IQ under 70, and then the majority of them are clustered around an average IQ of 80, which is above the threshold for ID. But their curve is shifted from the typical curve because of their CNV. So imagine again, in this case, the lower scores are more correlated with an intellectual disability, where higher scores on IQ, or the right end of the curve, are associated with the gifted IQ range. So here's our family again in blue, and let's say their starting point for IQ or intelligence measure is up there right in the middle of the range for the average population. So we've got the mom, dad, and the little boy in blue who have average intelligence, and then they also have a child with a CNV, and let's say this CNV can impact IQ. Well, in this case, it shifts that child's IQ from where it would have been, because it's very usually inherited around the same IQ within families, and it shifts it to such a degree that that child gets diagnosed with an intellectual disability or an IQ under 70. Well, let's take family B here. In family B, the starting point for this blue family is way up here, past the average range of IQ, highly intelligent families that have IQs that are much more toward the gifted range or on the high end of IQ. You have the exact same impact of that CNV on the child in orange, but in that case, it shifts it but doesn't end up falling in the range of intellectual disability. So we refer to that as shift. And shift is the impact of the CNV on certain domains, whether it's intelligence or social responsiveness or weight or motor abilities. And what is the impact of a CNV in those areas? And perhaps by looking at where the child would have been due to background genetics and other factors without the CNV, we can better predict where that child might end up and what the most need might be for that child. So in conclusion, family genetic background is known to contribute to variability in symptoms in all genetic conditions. So there's ranges. We know that family studies of quantitative traits, those traits that give you a nice curve and everybody in the population has them. So these are things like IQ and social abilities, language skills, motor skills, could allow more accurate prediction of prognosis in children with CNVs. So let's say now we're back to that 1-year-old who's just diagnosed with a 16p11.2 deletion, and the parents are wondering, what's the crystal ball telling us about where this child will be? Is he going to have schizophrenia? Is he going to have autism? Will he have intellectual disability? Will he be fine? Will he not really show a lot of symptoms? Maybe by doing family studies of this type, and also looking at all those environmental factors that we talked about, prematurity, medical conditions, etc., we might be able to better refine the prognosis and say, based on what we looked at, we think that this child actually has a pretty high risk for an intellectual disability. So we really want to beef up our early intervention for this child right from the beginning. In another child, it could be, this child may not actually have a lot of difficulties in the intellectual domain, but may have more social problems. Perhaps we want to say this is a child at particularly high risk for autism. Let's beef up those therapies that we know might help with social behavior. So our ability to more precisely predict the strengths and weaknesses, their vulnerabilities, or the things they're really good at in children with CNVs may allow more focused recommendations for intervention, because frankly, today what we're doing is saying, well, let's watch and see. Let's put them in early intervention and let's see where the areas of needs are. And, that works to some degree, but it would be much better if we could more customize based on the child's biggest areas of vulnerability. So ultimately, another reason why this is important is that we may need better ways of predicting prognosis in the future, because there's a lot of work being done right now on targeted pharmaceutical treatments that try for genetic disorders to correct or replace a gene product that's missing, for example, or somehow correct an overexpression of a gene product. And there's really a lot of work being done in that area right now. And ultimately, the goal may be, well, let's see if we can come up with ways to correct the genetic underpinnings. But if there's wide variability, which children would benefit most from a certain drug that helps with social responsiveness, for example, in 16P? Well, it might be those ones who really are at highest risk for autism. So knowing the prognosis and being able to more carefully customize treatment is really important now, but can also be important in the future. We have a long way to go before we get here, and a lot of the things we're thinking about are, we also don't want to stigmatize families or make them feel in any way that there's anything causative going on. And so really the idea is to look at lots of strengths and weaknesses, as I gave you the example of Michael Jordan and Lindsey Vonn. Maybe if you've got a CNV that impacts motor abilities, we can say in a family, your child may have some protection from the motor problems with the CNV because you guys actually score pretty high on the motor domain. So we really want to use this in a way to be able to customize interventions. I want to acknowledge our team out at Geisinger. We have our Autism and Developmental Medicine Institute team, which is out in Lewisburg, Pennsylvania. We also have an SVIP team that works out of Geisinger's main hospital in Danville, including Bethany Smith-Packard, Heather Rocha, Kara Filson, Simone Smith, who may be well known to all of you families out there. I want to acknowledge our collaborators at Simon's VIP. As part of the project that we're doing looking at DNA and looking at these quantitative traits at Geisinger, we have collaborators at Penn State University and also a team at Children's Hospital of Philadelphia. We have grant support from Simons Foundation and the National Institute of Mental Health. And of course, we would never be able to do this research or have an interest in this research if it wasn't for the many families out there who are contributing to the work and really drive us to dedicate our lives to researching these types of questions in CNVs and other genetic disorders. If you have any questions about this lecture, please send your questions to coordinator@simonsvipconnect.org, and those will be routed to me, and either I or someone else from our AVMI team will be able to respond to you. Thank you very much.